If you can't hear me, I think we should be fine, but I'm pretty casual about things, so just like raise your hand, even if you have a question or anything like that. First, I want to start talking about fish diversity. There are over 30,000 different species of fish, and this is actually comprises over half of all vertebrates on Earth. They're one of the most diverse groups that we work with. They have a huge variety of lifestyles and adaptations, diets, shapes, sizes, and ecological roles. Only 3,000 of these bony fish have been assessed by the IUCN Red List, and about 6% of those 3,000 species are listed as vulnerable or endangered. When we look at elasmobranchs, cartilaginous fishes, those about 30% of those are listed as endangered. So why do we care? Why are fish important? First of all, fisheries can provide jobs for up to 25 million people, and they account for about 16% of the protein that we consume as humans. They're a critical source of protein for impoverished countries. About 1 billion people in impoverished countries get protein from fish alone, and they're their sole protein source. Fish are the target of a lot of tourism and income mechanisms for a lot of different locations and countries. And hobbyists like myself just can't seem to live without them. But most importantly, they are critical in maintaining ecological balance. And they play many, many roles in the oceans, in streams, and rivers, and seas. They're super important for maintaining ecology, but we don't know anything about the microbes that live in and on them. So all microscopic organisms living in and on an animal collectively are called the microbiome, and this includes bacteria. And the microbiome, which I have represented here in green dots. Oh, I have a little laser pointer, I forgot about it. So the microbiome in these little green dots. Here, Chris, you get to dig through my bag and find my laser pointer. And they help maintain a happy and healthy host. And in this case, our little happy, healthy host is a little happy shark. So they can protect the host against potential pathogens. They can aid in breaking down food. They can provide the host with metabolites that the host may not otherwise be able to produce. They can be good indicators as to host health, especially with animals that don't usually show signs of illness. And they can also indicate the onset. Oh, look. Thank you. They can also indicate the onset of illness before there are any signs shown by the animal as well. And the microbiome is super important to priming the immune system early on in life. So how do we study the bacteria on these organisms? One particular method is called 16S barcoding. So let's say we have a community of bacteria. We extract the DNA from this community of bacteria, which is what this is right here. Each one of these bacteria has a gene called the 16S gene, and this is what we use to identify it. We then make millions of copies of this 16S gene from that community, and we sequence all of those 16S genes, and then we compare those sequences to a database so that we can then identify who's in that bacterial community. Another method that we can use is metagenomics. So this method will take all of the DNA from the bacteria and break it up into little tiny pieces. And it will put it all into a pool of fragments. And then those fragments are sequenced. And then we can align those fragments and collapse them into one big long sequence that's a consensus sequence. And then we can look in our database and see what those sequences are And then we can identify the genes that we're working with so we can get an idea of their metabolic potential. So these are the two methods that I'm primarily going to talk about today. 16S, there's a lot of advantages to 16S. It's a heck of a lot cheaper. You can identify thousands and thousands of species at once. However, you can only identify species. You can't tell what that microbe might be doing. Their genetic potential, you don't see into their genetic potential. You only get that barcode ID. It is quick and easy, relatively. And this is what the majority of fish studies use or have used. Metagenomics is very expensive. It's monetarily expensive. It's computationally expensive. It's labor intensive. But you get to look at the entire genomes of these bacteria so you can really understand what they might be doing and how they're interacting with their host. Unfortunately, not very many studies do this because it is expensive and it's hard to do. So in 2015, when I started this work, this was just about the most comprehensive fish microbiome paper out there, with 12 bony fish and 3 sharks, which out of 30,000 species is not terribly comprehensive. So this is their main figure. We can see that there are a lot of patterns that maybe you can pull out here. This tan bar here is the proteobacteria, and you will see the proteobacteria pop up over and over and over. There are also firmicutes that are in orange, and Fusobacteria in yellow, and what's the other one? Spirochetes in teal. Those are all organisms that you'll see popping up over and over. But you can see that these patterns are kind of hard to discern. There's only a handful of species of fish. Some of them only have 2 replicates. And there's a high amount of variability between all of these samples, which really makes microbiome work challenging. You need more samples and you need more species. So this work actually came out of our own lab. One of our graduate students said, there's nothing that looks at the life stages of different fishes. And fish have huge differences in their morphology as they age. And it's almost inherent that there have to be bacterial changes, but none of that was documented. And so he went in and he looked at damselfish and cardinalfish. And so reef fish, the way that their young life stage works is they have a pelagic stage where they are out in the open ocean just floating around. And when they get old enough and when they get close to a reef, they then go onto the reef and settle onto the reef, which is called recruitment, where they find a little home and they stay in that home. And so he wanted to look at larval fish before and after this recruitment phase. And this was an important paper because he was able to show that fish have a different microbiome before, which is the pre-settlement here, and after settlement. So they're having this shift in microbiome at their larval stage depending on whether they're still pelagic or if they've settled. And he pulled out some of the important players. Imagine that. We have gamma proteobacteria. This time we get down to Vibrionaceae. And also Endozoicomonas, which is interesting if you're a coral person because we actually see Endozoicomonas in coral a lot as well. So this particular study was the first of its kind. It's actually the first metagenomic study of sharks and actually the only metagenomic study of the bacteria on sharks, of the common thresher shark. And so once again, we see a lot of proteobacteria here. Imagine that. And this study is useful because we can start to get an insight of what interactions those bacteria might be having with their host, the thresher shark, and what they might be doing on the skin of the shark. But this study is also limited in its usefulness because you can see they only have 6 individuals here. Again, metagenomics is expensive, so getting more than 6 individuals is hard to do. And the only other reference that they have for these individuals is the water, which are these samples on this side. And so this is the first study of its kind, so it's very exciting, but at the same time, I look at this study and I'm like, well, what does it tell us? because we have nothing to compare it to. We have no other shark species to compare it to. It's different than the water, which they showed, which is very important because actually sharks aren't as slimy as you might think. And so we weren't even sure that the microbes would look different than the water, and they do, which is nice. But the study doesn't tell us a whole lot more than that. So the studies that I've showed you basically encompass the state of the literature, but there are so many other species out there. There's so many other lifestyles out there. And so this was the state of the field when I was hired as a postdoc at Georgia Tech to do fish microbiome work. And I very quickly realized that if I wanted to do fish microbiome work, I needed to come up with a comprehensive study. Otherwise, I would have nothing to base my hypotheses off of. So I did what any perfectly logical person would do, and I begged my boss to buy me a spear gun with university money, and then I took off into the middle of the ocean. So my study site was Moorea, which is the sister island to Tahiti. There are actually two field sites, or two field stations on Moorea. And I basically spent a month spearfishing in Moorea. And I went all around the island to these little grayed out dots, so all locations and all habitats of the island. And I essentially collected as many fish as I could from as many families as I could from as many lifestyles and diets as I could. So I collected 399 different fish from 15 different fish families from 53 different species all over the island. And yes, those are shark teeth, and I did rescue that sample from a shark. So we also had a Crest Net. And so if you'll remember what I told you about recruits, how they come into the reef. So this Crest Net catches all of those recruits that are coming from the open ocean and settling down into the reef. And so the crest of the reef is essentially right here. And this net sits on top of the reef. And these fish that come in overnight, they get swept into that Crest Net, and there's a little bucket here at the end that you can't see that we would go collect first thing in the morning, haul it out, and then assess our catch. So this is a typical Crest Net catch. You can see the diversity of recruits that we find. There's all kinds of shapes and sizes and species. This one is a little angelfish up here. These are two little baby butterfly fish. That's a squirrel fish. These are soldier fish. And I actually managed to get some adults from the Crest Net as well. These little squirrel fish figured out that they had a smorgasbord of food inside those Crest Nets. And so I counted like 17 little fish out of one of their bellies at one point. And they're not that big. They're like this little, yeah. So they thought they were so clever stealing my samples, but then I got adult squirrel fish out of that. So also, these little fish kept showing up, and I didn't know what they were. And they were live because they were still in the bucket. And I was holding one in a pool of water in my hand one time. They're very small. They're like this big. And I was holding it in my hand, and I watched it rear up and just chomp down on my finger. And my finger just started bleeding profusely. So fang bunnies make their living out of actually living in holes, darting after fish, taking chunks out of their fins, and then running back to their hole. That's how I learned what a fang bunny is. All right, so this is the big picture of my Moorea data. And so I took the gills of all these fish. I took fecal samples from the intestines of all these fish, from the recruits and from the adults alike. I also took in a bunch of environmental samples because I wanted to see if microbes from the environment matched up to the microbes we were finding in the gills and the intestines of these fish. And the big takeaway from this particular figure is that you can see there's a clustering of recruits, which is found in these pink samples, and then a clustering of adults, which is in the blue samples. Keep in mind, like all diet types, all species, this is a huge picture of 399 different fish. And recruits and adults have a very clear separation, so their microbiomes are very distinct, despite species, despite diet, despite everything. That seems to be a very big driver of microbiome composition. You can also see that gills and intestines have a little bit of a clustering pattern, which I'll show you later, but gills are hollow and intestines are solid. So I broke things down a little bit so we might be able to see some trends in diet. And indeed, surprise, surprise, microbiomes are dependent on what you eat. And so up here, the adult intestines, we see this nice little clustering of carnivores. And then we see herbivores up here, omnivores in between, with some carnivores scattered within. I think what's happening with those particular carnivores is that it's very, very hard to actually classify a fish as carnivore, omnivore, herbivore. And I found this out the hard way because people see what they eat once and they ate a fish, therefore it's a carnivore. But fish have a very different environment than we do, and they tend to just eat whatever is floating around in their face or whatever is in the sediment, which could be a combination of algae and worms. I think their diet is a lot more variable than we give it credit to. And I suspect that these carnivores that are classified in the literature as carnivores are actually closer to omnivores. We don't see any trends according to diet type in the gills, not terribly surprising. We see trends, they're not as strong in the intestines with diet type with the recruits. Again, probably because it's very hard to classify what these little bitty tiny fish are eating, if anything. So there's a lot of taxa that I found in over 399 fish, but we did find differences in the recruits and the adults. So the few I want to point out, this one that I can never pronounce, the brevinemytae, they're potential pathogens. So we did actually find more potential pathogens in the young recruits than in the adults. And then beta proteobacteria tended to be much higher in adults. All right, so we found that alpha diversity, which is on this side of the plot, alpha diversity is quite low associated with these animals, and especially within the intestines. We get some very low alpha diversity. However, beta diversity, which is basically the diversity between the different sample types, so between different fish, was much, much higher, meaning that individual fish basically had their own fingerprint of a microbiome. So I was sifting through all this data, and I told my boss, I think that actually I'm finding some of the same microbes from one fish in the gills as I am in the intestines. But I didn't know how to prove it. And he said, well, why don't you compare the diversity differences between the gut and the intestines of one individual fish to everybody else? And I did, and sure enough, I found that there are actually quite a few similarities between the microbiome of the gills and the gut of that same fish. Meaning that that one fish probably is secreting certain properties in their mucus that is cultivating their own individual microbiome on mucosal surfaces, whether it's on the gills or in the intestines. So this study landed on the cover of AEM, and it was really important actually for setting the stage for a solid future in fish microbiome work so that people had references to look at, and they could look at 53 different species of fish and say, okay, what does my fish look like? Or does it look like any of these fish? And what are the commonalities between the two? And so then I could actually have a little bit of fun and do some hypothesis-driven science. And I wanted to see, how does association with a sea anemone affect the clownfish microbiome? So basically, the clownfish is always in the anemone. It's got to do something to its skin microbiome, right? So what I did was I had 12 different fish, and the first day, none of them got a clownfish, or the first week. The second week, 6 of them got an anemone. So 6 of the clownfish got an anemone, and then 6 of them were forced to just be them by themselves. And this was maintained for about 6 weeks, and we took microbiome samples from their skin for 6 weeks. And then at the end, we took away all the anemones and wanted to see how the microbiome recovered. So we have the first week here where nobody has an anemone. Obviously, the 2 different types of fish don't look any different. But week 2, as soon as we let them snuggle up with anemones, we start to see a difference between those fish that have an anemone and those fish that don't, which is considered hosting and non-hosting. So we see these non-hosting fish here and the hosting fish here. There's a very, very clear partitioning between the two types in week 3. And they mostly maintain that difference throughout the entire experiment. But when you take away their anemone, their microbiome goes back to looking like a non-hosting fish.
Is it significant that in week 2 and week 3 the distribution kind of flipped, or is that not significant?
It's not significant because the way that you view an NMDS is that you could be viewing it from behind, you could be viewing it from in front. It's a multi-dimensional plot. Yeah. So I dug a little deeper and tried to figure out what was driving the differences between these anemones or these clownfish that had an anemone and which ones didn't. And there were basically 2 different types of bacteria. One was a Rubertalia, and it was indeed more abundant on the anemone, so it makes sense. It's rubbing up on the anemone constantly, then it's going to have more of that in its own skin microbiome. And then this other one could not classify for the life of me, which was the bane of every microbiologist's existence. However, this one seems to be increasing on its own. It's not super abundant in the anemone, and we don't know why. It could be that something about the anemone encourages it to grow. The clownfish is secreting something encouraging it to grow. We have no idea. So around the same time, a graduate student of ours was also exploring the clownfish anemone, well, not anemone, the clownfish microbiome. I didn't do anything but help feed the fish in this particular one. But he wanted to see how feeding a clownfish can change its gut microbiome. So basically, he set up this elaborate experiment of feeding fish at different times for about 30 days. And then he tested unfed fish and fed fish and whether or not the fish were fed every 3 days or whether they were fed 3 times a day. Turns out none of that matters except for whether or not the fish had eaten in the past hour. So unfed fish look like this. Fed fish, no matter how you fed them for the past 30 days and how complicated your feeding schedule is, they look like this. And so this is super important, though, because we find apparently that the gut microbiome can change very quickly. And lo and behold, the characters that are changing very quickly are Photobacterium, which is a gamma proteobacteria, which often pops up as Vibrio, which I've pointed out already to you guys, and Clostridium. So Josh also did a little bit of metagenomics, and again, metagenomics are very hard because it's such a jumble of data that often you can't get a comprehensive story out of it. But he was able to show that while fish were fed, there were genes associated with disease that would pop up, which is probably that Photobacterium. And then genes associated with metabolism were all in the unfed state before they were fed. So it was right around this time that Georgia Aquarium approached me and they said, so we're putting in a new shark exhibit and we need a microbiologist. Are you interested? I was like, of course, duh. We had already put out 2 publications with them, although they had nothing to do with sharks directly. But we have 2 publications with them. The first one is tracking the microbiome of the water column and their biggest exhibit, Ocean Voyager, which turns out is really not as stable as you would think. And then we also characterized all of the bacteria in their denitrification tower, which is really cool if you're interested in sulfur-based microbial denitrification, but that's another story. So we had these publications with them. They said, we like how you guys work. You already know fish microbiomes. Will you do shark microbiome work for us? And that's a no-brainer. And so we started with eagle rays because they have a nice set of eagle rays in captivity at Georgia Aquarium. I think this one is Dan. And they all actually, except for Dan, they're all named after cereals, which is kind of funny. So yeah, my data set is like Kix and Cookie Crisp and Honey Nut and yeah. Yeah, and then Dan. I don't know what happened to Dan. So anyway, they have a collaboration also with Moat Marine Research Laboratory, and they also get samples from wild eagle rays as well. So this presented a nice study for us to dive into the elasmobranch microbiome work. And this also provided a master's thesis for one of our students. So you'll have to forgive the resolution because these are the only pictures of hers I could find. But here she has plotted all of the microbiome samples from both wild and captive. She hasn't differentiated that yet. So we can see that the gills and the skin are relatively similar. They cluster with the water here. But the cloaca, which can or cannot be indicative of their fecal samples, are very, very diverse. Just like we saw in the fishes, they probably have their own, each have their own individual microbiome fingerprint within their intestines. So here she's looking at the gills, and we're looking at wild, which are these blue, and then OV, which is ocean voyager. I'll say that a lot. That's our captive population. And we can see a very, very distinct difference between the two. They are very different, and they're being driven by the water. And we know this because our water samples are clustering here, and each of the gill samples are clustering with the water. So same trend with the skin. It's not quite as strong, but it's still pretty strong. We see the wild population cluster with its water, and the OV water more or less cluster with its water within itself. But our cloaca samples are all over the place. So this is telling us that there's probably something other than the water that's driving the differences between these individuals. As you can see, there's actually not a whole lot of clustering between wild and captive eagle rays. And actually, Georgia Aquarium was very excited to see this because this suggests that the diet that they're feeding their individuals are spot on and match what the wild individuals are eating. So once again, we look at alpha diversity, and we can see that alpha diversity is lower for the captive individuals in pretty much every niche. And this isn't terribly surprising because they filter that water like crazy and they bring down that diversity, and actually the microbial density is very, very low in that water as well. But once again, our cloacas have very, very low alpha diversity. Photobacterium, imagine that. And so their cloacas are full of these Photobacterium. You see them both in the wild and in the captive. So we have captive down here, wild down here. The captive ones tend to have a little bit more Vibrio in them. Photobacterium and Vibrio are very closely related, and they often get a bad rap as disease organisms, but I hope to prove by the end that that may not always be the case. So looking at the gills, remember I told you that the gills were driven by the water. So we know from our previous study when we looked at the microbes in the water column of Ocean Voyager, Cordymonas is always there, and it's one of two bacteria that actually make up about 30 to 50% of the bacteria in the water. And so in this particular case, at the day that they took the samples from these particular eagle rays, it was about 16% of the bacterial community. It can be much higher. And so we know that there's the shared niche between the gills of these eagle rays and the water column. And this goes for the wild population as well, although it was smaller. Okay, so I've taken you guys from clownfish to eagle rays, but we haven't touched on the large predators yet, and the large predators obviously might be quite different. So we went from Georgia Tech to Georgia Aquarium, and then Georgia Aquarium said, hey, we have these collaborators called OCEARCH, and they're looking for a microbiologist. Are you guys interested? Of course I'm interested. So what OCEARCH does is they basically provide white sharks to scientists. Because white sharks turns out they're really, kind of hard to catch and hard to handle, hard to sample, and it also is very expensive. It costs a lot of boat time and a lot of gear time and a lot of people who really know what they're doing. Most scientists can't afford that. And so large predators such as white sharks, really in-depth research on these animals has been lacking because of that. And OCEARCH realized that, and they said, we want to fix this. And so they're a nonprofit, and they provide the boat time, they provide the animals, they bring the scientists on board, and they catch the animals, and then they say, here, sample our animals. And so we had been receiving samples from OCEARCH for the past 3 expeditions. So they send us samples, actually, next slide. So 3 expeditions, there are 3 different expeditions that we've gotten samples from. And there are, let's see, 7 white sharks, 4 white sharks, and a handful of tiger sharks as well. And we get swabs from their skin, from their gills, from their cloaca. Sometimes we get fecal samples from them, so there's 2 fecal samples. So this is awesome because we have different sharks from different times and we have different locations and lots of different swabs from them. So we can actually start to get a picture of what the microbiome of a large predatory shark looks like. So if you go to the OCEARCH website, it's really kind of fun because they have all these little icons that you can click on. And they are sharks that are pinging that they've tagged. And so this is actually an individual that I have samples from. So this is a lot of fun to play with because for me it's like personal. And I particularly love the little jaunt through North Carolina here. Yeah. Yes. There was actually, well, there's a funny alligator one too. Okay, so we're particularly excited about these fecal samples because they're microbial gold, because we can actually get so much data out of these fecal samples. How do you collect a white shark fecal sample, you ask? Well, this is how. The next time you feel grumpy about your field work, just remember you're not the one holding the claspers aside. So they also do a variety of science. They take parasites off of the animals and analyze them. They insert acoustic tags. You can see the parasites on the shark fin right here. And these guys are drawing blood back here. So one animal serves for about 17 different science projects, of we're just one. Okay, so back to data. This is the microbiome data from the white sharks and tiger sharks that I just showed you. And so we have, it's kind of hard to see, I'm sure, from back there, but we have white sharks that are circles, tiger sharks that are triangles, and a few water samples that are squares. And so the big punchline of this particular plot on this side is that all the samples partition out into expedition. So remember I said there were 3 different expeditions. There were 3 different times they went out to catch these sharks from 3 different locations. And this is the Nova Scotia expedition, this is the Gulf Stream expedition, and this is the Southeast expedition. So they're not partitioning according to species. They're not partitioning according to cloaca, skin, or gill. It's entirely on what expedition that they came from. So if you look at these 2 graphs, this is unweighted unifrac and this is weighted unifrac. As soon as you look at weighted unifrac, they start piling on top of each other and they all look the same. So this is telling me it's probably rare microbial species that are driving the differences between these 3 expeditions. So I pulled out the one expedition that has both white sharks and tiger sharks because I thought there would be a really interesting interspecies comparison. Turns out nothing's going on. There's no difference between these triangles and circles, which is interesting in itself because actually tiger sharks are one exception and they are very slimy, and white sharks are not very slimy at all. They're very rough, and yet I can't distinguish their microbiome, even in the cloaca. So that's actually interesting to me, how much the environment influences their microbiome. So as I said before, people were entirely convinced that their skin microbiome was going to be different than the water microbiome, especially after I just told you that the water microbiome is so influential on their own microbiome. But in fact, and we can just look at this top row up here. So I tested each individual bacteria to see if it was significantly different between the water column and the sample that it came from. And it turns out there are, from each sample, from 29 to 42 different bacteria that are significantly enriched in the gills or the skin or the cloaca of these animals. That's not as high of a number as you would expect to see from other organisms, but it's also not the same as the water. So they are still harboring their own microbiome. So alpha diversity, once again, and I'm just going to point out this crazy low alpha diversity for the fecal samples, once again. Not a whole lot of difference between tiger sharks and white sharks, again. So my boss and I started looking at this low fecal microbiome, and we said, all right, this seems to be a trend. We've seen this a lot by now. So let's dig into this a little bit more. So what I did was I took data back from Morea, from my fish survey, and then also from a handful of other studies that have been published. And I'm just looking at number of SVs, which is basically the number of different bacteria that I find in fecal samples. And so we can start up here. These stegastes, these farmer fish, have like over 600 different types of bacteria in their gut. These are herbivores. We can see also these are land herbivores up here. They're higher, not as high as these stegastes, but they're up there. We have toothed whales here, and they're lower, but they're still not crazy low. With there's a few exceptions in there, but that data kind of looked kind of funny, so I'm not sure that is reputable. But you come down to here to these white sharks, and there's only 11 different types of bacteria found in the guts of white sharks, which is just crazy low. My boss made me redo it like 10 times just to make sure. Turns out it's true. And so we kind of see, I also pulled data from this particular fish, the arc-eye hawkfish, because it explicitly eats fish, and I know this, well, fish and crabs, but it doesn't accidentally eat any seagrass or anything like that. I know this. I've been in the guts of like 30 of them. So I'm confident that those are as close as I'm going to get to a white shark of a fish version, of a teleos fish version. And you can also see that they are also quite low, although not as low as the white shark. So this is actually what their diversity looks like, and you can count them. There's all of like 7 taxa listed here. And these are 2 different sharks from 2 very different locations. And their microbiome is crazy similar. So this blue is the exact same Photobacterium. This gray here is Clostridia, as well as this orange. So they have a slightly different Clostridium. They have the same Campylobacter, which is this yellow. And then they have this same, another Clostridium. So they have a lot of the same taxa, even though these sharks were caught like 1,000 miles apart. So actually this is really exciting for me because this means that I can do metagenome work on these fecal samples quite easily, which is not found very often. So I took these samples and I did metagenomic sequencing on them, and I actually managed to isolate 5 different bacteria genomes from these fecal samples. And actually, many of them are very high quality. One of them is 95% complete, and actually no Photobacterium gets more than 95% complete, just because of the way that's calculated. So this is probably a closed genome at this point. So I'm super excited about that I've got a couple of genomes out of these shark fecal samples. And I'm particularly interested in these Photobacterium genomes. So I've mentioned Photobacterium a lot. And so this is a family tree of all genomes of Photobacterium that have been sequenced. My two are right here. And this is a little concerning to me because these are all actually listed as pathogens. And those are the only Photobacterium that are in any of GenBank or any sequenced Photobacterium. So I've already shown you that pretty much every fish and every shark is full of Photobacterium. There's no way that every single one of these sharks and every single one of these fish is sick. That's just not possible. But yet, anytime anyone sees Photobacterium, they're like, my fish is sick. This fish is sick. I've reviewed so many papers that say, we found Photobacterium, this whole population is sick. And that's just not possible. And so I think that Photobacterium is basically like our E. coli. We all have it in our gut. For the most part, we all need it. Every once in a while, there becomes a pathogenic strain that gets around and makes people sick, and then people freak out about E. coli. But realistically, it's in everybody, and everybody needs it, and it's not uncommon. But Photobacterium and fish gut microbiome is so new that nobody understands this. And so I think it's particularly important to study this at the moment because we're getting aquaculture people that are going, Photobacterium everywhere, everything's sick, sterilize everything. And that is not the answer, not by any means. So I hopefully am going to dig into a little bit more of these Photobacterium genomes, and I want to understand what makes a Photobacterium pathogenic and what makes it non-pathogenic. Can we understand the difference? Can we test for the difference? Are Photobacterium essential to fish health? It seems like they are, since they're found in every single fish. And are they host species specific? Are there individual Photobacterium strains that are found in specific species of fish? And what does that mean? Did they evolve together? Do they happen to just share Photobacterium? Can Photobacterium go anywhere? I think these are really important questions to understanding fish health. So, I hope I've shown you that carnivorous fish, particularly the top predators such as white sharks, they seem to have a remarkably low fecal microbiome diversity, and we don't really know what that means yet. We don't know if it means the animal might be more prone to sickness, if they just don't eat very often, so therefore they can't support a large diversity within their gut. They might have a very high urea content, which might kill off a lot of microbes. I don't really know. We'll find out. But we have also shown that they have the same basic structure from clownfish to white sharks, and that is Photobacterium, Clostridia, Fusobacterium, and Campylobacter. Those are pretty much the four staples of every fish microbiome that I have yet encountered, which is a lot. There are many factors that also influence the fish microbiome, some of them which are intuitive, some of them which are not. It depends on who and what you're rubbing up against. It depends on your particular species and the life stage. It depends on whether or not you're captive or wild, or more likely the water source that you're being subject to. And also it depends on what you're eating or not eating. So we have a lot of projects in the works at the moment. And so obviously we are in collaboration with Georgia Aquarium, and we now share a graduate student with Georgia Aquarium. And he's working on his PhD in whale shark ecology and whale shark microbiome. And so he has had the pleasure of sticking his hand all the way up a whale shark gill. And so we also have this really complex data set as well that is just full of sharks, and we're still trying to figure out what to do with it. But we have tiger shark, or not tiger sharks, that's a zebra shark, and we have great hammerheads and silkies and sandbars and sand tigers and black tips. And so we've accumulated this massive elasmobranch microbiome data set that we hope can be essentially the same thing that I did for the reef fish, was create a baseline of all different species, of all different lifestyles, so that we can start understanding what actually is influencing that microbiome, so we can start to understand the fish health and what's keeping them healthy and what might not be keeping them healthy. So I have a lot of people to thank, because obviously this is a very massive project. People from OCEARCH and from Georgia Aquarium, especially Al Dove and Lisa Hoops, and my PI, who lets me run around and pretty much do whatever I want, as you saw. And then our graduate students, a lot of which have done a lot of this work. So I'm ending a little early because I'm expecting some questions from this.